The barriers to making antibody-mediated prevention of HIV were very well established. The wind behind the antibody development got stronger and stronger.
It became very obvious that we were going to have to induce a broadly neutralizing antibody vaccine.
It's really changed how we're approaching HIV vaccine development and gives us new excitement and new energy around the possibility.
The HIV vaccine field is at an interesting sort of viewpoint. The outside world says, gee, can you guys ever do this? You know, there's a sort of a dourness. Can you succeed?
If you're going to vaccinate somebody against a highly variable virus like HIV, you can't just induce neutralizing antibodies against one strain, because there are literally hundreds of thousands of different strains that are circulating.
We have two approaches that could be used to try to provide protection. One is to just give the broadly neutralizing antibodies themselves as an infusion, and that might provide protection. It also gave us a target to shoot for developing vaccines that develop broadly neutralizing antibodies.
We really brought forward this a rational approach, germline targeting it's called, to generating protective antibodies against HIV.
There are a number of these germline targeting vaccines and other vaccines that are designed to generate broadly neutralizing antibody responses going into human trials. And these vaccines have all been very safe.
I think that the most recent clinical studies showing that we can guide the immune system to a series of immunizations to start to make really good antibodies and really specific immune responses. Those are very encouraging.
We're targeting antibodies that people are able to make, because these broadly neutralizing antibodies have all been recovered from individuals who are living with HIV and have developed these. So it is more of a pathway that the immune system is able to take.
But still, it took 4 or 5 years in an HIV-infected individual to make broad neutralizing antibodies. So our task became, over the years, how do you induce broad neutralizing antibodies?
And actually, in the last year, we have, evidence that we can prime these B cells that we want that will eventually lead to mature antibodies, and that we can, vaccinate with a different vaccine and move them along.
We and other members in the field have started to make a lot of progress with either the beginning of the immunogens, the beginning of something to hit the naive B cell receptor and start it off.
We know how we can analyze the data, but we really need to be able to be very creative in developing the immunogens that will help us get there.
We've gotten to as far as they're sort of in early elementary school. We haven't got them to college yet. But the data are suggesting that the processes that we're doing are being successful. So with harder work and a little bit more empiricism, we should be able to, educate them enough to make mature B cells.
And what we've been able to do is with the IMPER to get it all the way to heterologous neutralization. And now we're working on how do you get it to be more broad and potent. Well, that's a real big beachhead for us.
You need an ID80 of 200 against that virus to be protected from acquiring infection by that variant. That's the metric that is now used for designing the next AMP trial. We now know that's the level of neutralizing activity that we need to achieve and maintain over time in order to have really great efficacy.
So we are spending our time and energy trying to make one bNAb at this part or that part. But to make a vaccine, we actually need to put them all together so that you make the immune responses against 2 or 3 or 4. There's one vaccine that will prime the germlines of 4 different bNAbs. And there'll be another vaccine that boosts those 4, and then another vaccine that educates them, to junior high, and the last one that'll get them to go to college.
Now that we know what that bar is, we know what our goal is. We know what we're shooting for. We didn't have that information before, and it's really useful information to have.
A big door to the idea that vaccines that are dependent on antibody are a credible and probably the best strategy for the ultimate generation of an HIV vaccine.
It's an enormously complex disease, and thank goodness we have good antiretroviral drugs that work.
I'm very optimistic that substantial progress is going to be made in the next 5 years. That is why I'm not retiring. I am 70. I'm going to keep going.
We will get there when the science teaches us how we can program the cells to do what they need to do. I see the technologies of vaccines really moving at a neat pace.
I'm also hopeful that eventually there will be a vaccine or very long passive immunity for the highest risk people.
We have a roadmap for how to do it. We just have to be successful.
I see the pathway to a vaccine. I hope that we can remain committed to it.
It's a very exciting time. I want to be a part of it, and I want to continue to contribute to this effort.